Good afternoon. Welcome to DESAM Research Institute webinar series. We are having the third COVID-19 related webinar in 2020. Today we have two international speakers to talk about SARS-CoV-2 genome analyzing. As you are aware of, understanding of the SARS-CoV-2 genome is not only necessary for the development of new treatments and vaccines, but can also be used to improve the sensitivity and specificity of existing approaches for virus detection. It is important to understand SARS-CoV-2 genetics, genetic diversity, and reported mutations, molecular-based diagnostic tests, and genetically related vaccination strategies. You can ask your questions at this chat section, and our speakers will answer them just after their talks. Before inviting the first speaker of today, I would like to give a brief information about him. Dr. Ewan Harrison did his PhD on bacterial genomics at the University of Leicester. He has followed by postdoctoral research at the University of Cambridge, where he investigated the transmission of bacterial pathogens between human infection and zoonosis. He is now a UKRI Innovation Fellow at the Wellcome Sanger Institute at University of Cambridge. Dr. Harrison is a microbiologist who uses a combination of cohort studies, bacterial genome, and human genomics to investigate bacterial pathogens. Since March 2020, he has been seconded to COG-UK, where he coordinated the setup and now acts as a deputy director. Dr. Ewan, hello. The platform is yours.
Hi, hello everyone. I'll just share my screen. Okay, can you see me? Is the screen? Is it?
Yes,
visible?
there.
Yeah. Okay. Thanks. So it's a real pleasure to come and talk to you today on behalf of the COVID-19 Genomics Consortium in the UK. So what is COG-UK? So we're a 17 site sequencing network that was set up in basically March last year. A group of academics, scientists in the UK, along with the four public health agencies, came together to discuss really the possibility of, as the SARS-CoV-2 pandemic was starting really in the UK, that whether or not we should be leveraging our sequencing capacity. So the sequencing network came together, and very, very quickly we put together a funding application to the UK government, which we received on the 1st of April last year, and that gave us £20 million of funding to set up this network. We have 16 regional sites, as you can see here, across the UK, and we also have a large central sequencing hub at the Wellcome Sanger Institute where I work, which is obviously has a really huge sequencing capacity. The COG network is administrated out of the University of Cambridge, and Professor Sharon Peacock is our director. So what were the objectives? So in our original funding application, we outlined really four major objectives of our sequencing program. One was to generate data to detect mutations and variants. Two was to generate data really to track virus spread and to support outbreak investigation. We've also been working to combine data sets. So we've been doing this more recently, and I'll talk a little bit about how that's quite important. And we're also now working to combine data sets, so we'll have both human genomes and viral genomes that will allow analysis of both. And we're also keen to support national and international collaborations. And we're now really starting to think about all the things that we've learned over the last year and starting to get those on, published and out there for other people to make use of. So it's been an evolving project, really. As I said, at the beginning of April last year, we stood up a network, and very, very quickly, with frenetic speed, put together a network that worked. As of November last year, we received more funding, and we've started to scale up our sequencing capacity now. So we can sequence over 20,000 SARS-CoV-2 genomes a week. And we're also starting to implement this as part of a national sequencing capacity that will be handed over to the UK public health agencies. And we're now in that stage there where we're beginning to there's going to be, within our public health agencies, the capacity that we've built as an academic sequencing network is now going to be handed over to the government, and then that will exist for the long-term future of pathogen genomics in the UK. So it's been an ongoing process. How do we work? Well, we have a reasonably complex network. Within the UK, diagnostic testing happens in 2 locations. So if you're out in the community and you get a SARS, you think you have COVID-19 and you have symptoms, your sample will go to one of the large UK Lighthouse Labs network, and these are really huge diagnostic testing laboratories that can deal with, they deal with tens of thousands of samples a day. If you get sick and you obviously, you have more severe COVID-19, you'll end up in hospital, and then our testing samples go through normal NHS diagnostic testing labs, which are all over the country. Broadly, how our network operates is that these mass testing samples flow to the Wellcome Sanger Institute, and also now to a couple of other sequencing sites. And all our NHS, so this is the patients, this is people with severe COVID, all their samples end up in one of our regional sequencing sites, the 16 that are located over the country. We've stood up a combined computational infrastructure, which makes use of the MRC cloud infrastructure, which was a really critical component of our ability to stand up a sequencing network, because we had this existing computational capacity that we could build on. And within there, we have quality control and analysis pipelines, and also some limited metadata flows into there as well. And I'll talk into a bit more detail about that. And then our data flows straight into the UK public health agencies, where it's now used on a daily basis for horizon scanning, for identification of variants, but also for investigating outbreaks. And it's also been fed back to the hospitals themselves, but also it goes to our wider sequencing network, where we have a large number of academic scientists who are also making use of the data. And I'll talk a little bit about some of the work that they've done. So as I mentioned, in terms of hospitals, there's a huge number of NHS hospitals that are all over the UK. And you can see here by the coloured dots, we have sequencing sites all over the country, and then clusters of hospitals send their samples in centrally. So for example, you take all the blue ones here, they all go into Newcastle here. So we have quite a large network, and there's obviously a huge logistical operation that has to happen each day. The samples have to flow into this central sequencing hub. As I mentioned, we have this big network of sequencing diagnostic sites called the UK Lighthouse Labs. So these now have actually expanded. There's more than in this initial map. But these send all their samples to the Wellcome Sanger Institute. So that the Sangers had to stand up a really massive logistical operation. So they're receiving over 500,000 samples a week. They come as deep well 96 well plates like this. There's a huge now freezer container park that's been built, you can see here, that takes all those samples, and there's capacity for storage of over 6 million samples on site. They then go into robotic area cherry picking, and at the moment, basically all the positives that are coming in each week are being picked into new plates, and then they go on to be sequenced on NovaSeq at the Sanger Institute. And in times of well, there's huge numbers of samples, the excess then goes up to the rest of our sequencing network. But at the moment, the Sanger is capable of sequencing all the community testing samples it receives. So how do we do the sequencing in the UK? Well, one of the foundations of the work that we've built is the Arctic network, which was developed actually, obviously before COVID, and it was developed for sequencing emergent viral pathogens. So they very quickly developed the Arctic protocol, which has been adopted worldwide. And across our network, we use both Oxford Nanopore. We also use various Illumina platforms, and also Bio. And we have a number of different protocols that people are running. We call our protocols open access, and we're currently constantly working to reduce costs and improve the plexing, the throughput of the samples that can go through. We've seen a massive reduction in costs over throughout the pandemic. I think now the cost per sample are just over £10, where at the beginning of the pandemic they were £50, 50 or 60 euros. So there's been an ongoing research and development effort. So as I mentioned, we have a central computational framework, and this has been really critical to our success. It's described in a bioRxiv paper here by Sam Nichols, who developed a lot of this basically computational infrastructure. So it's based out of the universities of Birmingham and Cardiff, and this basically takes all our sequencing data every day from all the sites across the network. It then runs it through, matches it up to patient metadata, so things like the date of sampling and the location of the individual. They then have a quality control and phylogenetics pipelines that run daily. It also has an automated upload to the European Nucleotide Archive and GISAID, and it's also a secure system that allows us to handle patient level information all in one place. And this is really critical because it allows us to have all the UK data in one place. And we had to stand up a particularly complex legal framework to allow us to do this for the first time, because normally the 4 nations of the UK don't share this data. So that's been really critical to our success. So as a network, we've tried to adopt an open science and data sharing policy. So as I mentioned, all our protocols are online on our website. We also make all our data publicly available, basically within as soon as we possibly can. So that either goes, as I mentioned, into GISAID, it goes into the European Nucleotide Archive. It also goes on our website, and actually at first, before we could get any of these to work, within the first week or so, we make all our data available on the website. We also have a number of tools that allow you to interact with our data and visualize it. And we've also been making all the software that we've been developing open access and available for people to use as well. So we have we've implemented since quite early on a sampling strategy that basically splits our sequencing in two portions. 50% of our sequencing is nationwide surveillance, and we try to do this in a random fashion. So you can see here, this is some coverage for the UK on the 1st of March, and you can see, although there is some variation in the coverage, we do have coverage across reasonably equally across the country. This goes on day in, day out. We never change this community sampling, so this ensures that we have a snapshot of the viral populations across the UK. The other 50% of our sequencing is targeted, and this is towards patients with severe disease. We're now tracking any cases of vaccine failure, reinfections. We also have anybody who reports a travel history. We get their samples and they're sequenced. And there's also been large-scale enrichment studies. So this is where a variant of concern has been detected in a particular area. There's been mass sampling in those locations, and we've been sequencing those. We also have a number of national studies within the UK. So we have a couple of surveillance studies that operate each week, run by the Office of National Statistics, and another one called REACT. We've also been sequencing vaccine trials, and as I mentioned, we also have an interest in the human genome as well. So we're trying to ensure the genomic study, which targets people with severe and mild COVID-19 disease, having their genome sequence, we're capturing viral sequences. This has resulted in us sequencing about 400,000 viruses at the moment, and it's about, I think now it's about 9% of cases we've managed to sequence since the beginning of the pandemic. And because case numbers in the UK are now quite low, we're sequencing over 40% of positives. So one of the critical things that we, as I mentioned, we try to do is to ensure global access to the data that we've generated. I know this has been globally. There's been problems with this with data sharing, because obviously some of this contains potentially patient identifiable information. So from early on, we've agreed that we will only release the sequencing data along with our central sequencing ID, the date of sampling, and the UK nation. This means that basically there's very limited risk of deductive disclosure. So that is people using this information that's available to find out something about the individual patients. There's no controls on this data. So basically, once it's in the public sphere, we have really no control over how people use our data. I mean, it is best practice people to ask and to acknowledge the data generation, but there's no guarantee of that. But we've decided that it's more important that the data is made publicly available. And I think as we'll go on to discuss, this is really critical. We also can have much more restricted data, so all the members of our consortium can access things like the outer postcodes. This is geographical information, the age and the sex of patients. And we also have a much more rich data set that's much more restricted, and you have to put an application into internally to access this data. And we think this is a good compromise of making sure that the data is publicly available, but also available to people for analysis. So one of the key things we're really focused on now is understanding COVID-19 disease through ensuring that sequencing data is integrated with other data sets. So as I mentioned, we've kind of got three particular things that we're interested in terms of severity of COVID-19 disease. So we've been targeting, as I said, severe COVID-19 cases, and we're now linking viral genotype information and lineage information to complex clinical records. We're also about to start analysing the host and viral genome, and we've got about at least 20% of the cases we think that are linked between the two, and we're working to enrich them. As I mentioned, particularly with the emergence of variants of concern, we're now doing in trusted research environments in the UK, which I'll go on to describe in a little bit more detail. We're linking the viral sequencing data with clinical records to look at how outcomes are affected and to take into consideration things like comorbidity to see how that might impact along with viral genotype. We're also interested, obviously, in immunity, so we're ensuring that any cases of vaccine failure are sequenced, and there's a study called the Siren study, which screens healthcare workers routinely who've been vaccinated to identify cases of vaccine failure, and that data is feeding into the government as they're monitoring particular variants on a day-to-day basis. That information may well be critical for risk assessment of a particular variant. We've also been concentrating on looking at transmission in hospitals, and I'll talk a little bit about some of the work that we've done in terms of transmission in hospitals and how we've been using genomics to track that. And as I also mentioned, the community transmission studies, we're now sequencing and integrating this with more rich demographic information to understand, things about how COVID-19 transmits in certain households and demographic groups. So one of the problems that we've faced and we've overcome is this linkage to clinical records. So as I mentioned, we make all our data publicly available, and we've now stood up a system that will link our publicly available data with individuals in UK public health agencies. And this will allow us to link data that's held in a number of databases. So there's one called ISARIC, which is a clinical cohorts of very severe COVID-19 cases, but also other trusted research environments in the UK. And these hold really rich patient-level data that can then be linked to viral genotype. And we're doing this by using the healthcare identifiers of individuals, and then we pass this data over, and it allows really detailed analysis of outcomes of COVID-19 disease linked with viral data. So I've thought now just talk a little bit about some of the things that we've been using this data for within the UK. So quite early on, we've been using, since basically day one, we've been using the data that's been generated across the UK to look at outbreaks in hospitals. It was quite interesting right at the start of the pandemic that in the UK, there was lots of people who thought that we weren't going to be able to use sequencing data from SARS-CoV-2. It wasn't going to be informative because of the low mutation rate compared to things like influenza and HIV. But actually, we found quite early on that sequencing was very powerful to identify outbreaks and investigations in care homes, etc. So one of the best examples was an outbreak in a hospital in Cambridge, Addenbrooke's Hospital, where I also work, and they identified that there were patients who were coming in for kidney dialysis who were getting COVID-19 disease, and they couldn't work out how the individuals were picking up COVID-19 disease despite the infection control practices were in place. When they combined the genomics with epidemiology, they identified that actually it was a shared transport, that was people were coming in a minibus to the clinics, and identified this was the common variable from the genomics that was bringing the patients in, and they were picking up COVID-19 on the transport. So they switched them all to coming in using taxis, and identified, and this stopped the spread of COVID-19. So without the genomics, they couldn't have paired those cases together. We've also done this at a national level. There was a study called COG-UK, which is the hospital onset COVID infection study, which ran over 3 months and has now been written up. And they sequenced every single case entering hospitals, a number of hospitals across the UK, and have assessed how well COVID-19 impacted on reducing hospital infections. So some of the important work that we've done, we tracked the importations into the UK, and a group led by Ollie Pires and Andrew Rambo analysed the introductions of COVID-19 into the UK during the first wave. And despite a lot of the focus on China at the time, this identified that actually that during the first wave, most of the importations into the UK actually came from France, Spain, and Italy, which really I think wasn't apparent at the time, and I think it's really identified that, you know, for future pandemics, the use of sequencing is going to be really critical for, you know, making assessments about border measures and other things about importations of cases. We've more recently followed this up by looking at introductions during the summer, once people could travel again. In the UK, there was a policy of that anybody who wasn't travelling to a country that had a travel corridor, so where you were allowed to travel without quarantine, you had to quarantine for 14 days. And we just completed a study where we were combining contact tracing data, so this is the information when people came back, when they were positive for COVID-19 disease, whether they had contacts, where they travelled from. This identified actually during last summer that over 50% of cases came from just 3 countries. So it was 20% from Greece, 16% from Croatia, and 14% from Spain. Of these that had a genome available, we looked to see how well this 14-day quarantine process actually stopped onward transmission. So by what we did is we identified individual cases that then were linked to further transmission using the genome. And we found that when people had been quarantining, it massively reduced the likelihood of them transmitting cases compared to countries where there were no travel restrictions, so you could move freely. We also identified that the 16 to 20 age group was responsible for a lot more of this onward transmission than was previously understood, so they can be targeted for public health messaging. But also critically, I think, with the importation of variants, even the 14-day quarantine didn't completely reduce the risk of onward transmission. So this is obviously important information for government when they're thinking about future controls on borders, etc. So we've also been tracking mutations in the UK. So this work now is published in Cell. Early on, when the D614G spike protein mutation was identified, because we were able to integrate our data from UK-wide, we were able to track the spread of strains of lineages with D614G. We were also able to look at outcome data, as I mentioned, by linking to the clinical records. We were able to assess whether there was any difference with this mutation on transmissibility, and then also ultimately pathogenicity. And it's estimated that this version is the D614G mutation does increase transmissibility. And these kind of analyses were really more important for the kind of the final piece of work I want to talk about, which is the identification of the B117 lineage in December 2020. And I think this really exemplified the importance of what had been built in the UK for detecting the emergence of viral lineages. So in early December, you can see here that the number of cases in the UK started to rise, and particularly these were targeted in the southeast of England, in the area around Kent. So because we were conducting the routine surveillance, so day in, day out, we were just sequencing random samples from across the area, we could look in those areas and we identified that there was a particular lineage that was overrepresented in the southeast of England. You can see here this is the lineage, and it had acquired 23 unique mutations. This obviously made everybody very quickly look up to see what was happening, because cases were increasing in this part of the country while they were resting elsewhere. This allowed us to identify that that the mutations also caused a dropout in one of the 3 targets in the TACPath assay, the diagnostic assay, that allowed us to track the increase of the B117 lineage, and you can see this is the actual data from the TAC path dropout. You can see there was a massive increase in F gene dropout. Again, combining, so this allowed the UK government, the public health agencies to report this within about 3 or 4 days of it being detected to the WHO, and allowed countries around the world to change. Obviously, there was border controls put in place for people travelling from the UK because of the risk of the emergence of this lineage. And as I mentioned, the linkage with the health record data has now allowed people to identify that this is associated with moderate increase in severity of disease, but also has been able to estimate that it's at least 40% more transmissible, which again has been really critical information. It was only by having a very rich genomic data set linked with the healthcare record enabled people to do this. So just to summarize, so really since in a matter of about 2 to 3 months, we stood up a UK-wide sequencing capacity. We've generated about 9% of all UK cases. We've built an integrated central computational system, which I think is really critical. And we've made a number of key scientific findings over the last year. We've tried to share our data as widely as possible, and we've hoped that that's helped others around the world. And we've tried to kind of act as to really set an example by doing that. And we're also obviously now working to kind of the lessons we've learned over the last year to share internationally. And we're working now really on as the public health agencies take over sequencing, we're working to ensure that our data is put to best use in academic research. I just have to finish with a slide that includes a few of the over 600 people who've been involved. There's probably about 400 people involved on a day-to-day basis at the moment. And I'm very happy to take any questions. Thanks.
Yes, Dr. Ewan Harrison, thank you very much. Actually, it's very nice to share because during this pandemic, it's very good to share, and it's going to be beneficial, and it has been beneficial for all over the world. I think that we can share the data that fast. I want to ask you a question. Are you just sequencing the UK cases, right? You don't get any the cases from like Commonwealth countries or in other countries?
No. There are. I'm not entirely sure what the government is talking about offering sequencing services, but I'm not sure entirely what the plans are at the moment for that.
Okay.
But we are talking to a number of countries around the world about who are thinking of standing up similar systems, and we're trying to provide as much advice as we can from our learned lessons.
Okay, and there is a question from Dr. Bas Oude Munnink. The UK variant was already circulating since November, if I am correct, picked up somewhat later, although still very fast. How can we try to detect this variant even earlier, say?
Okay, yeah, so I think the earliest case was actually in September, but obviously that was 1 or 2 cases. So now in the UK, there is basically daily meetings. So they're monitoring the data. So at the time that wasn't stood up, but we're monitoring it on a daily basis. So the UK public health agencies and academics, including a number of the people involved in our consortium and other virologists, etc., meet on a regular basis to look systematically. So we're also basically we've got systems in place now that look for the emergence of particular lineages that are growing faster compared to others, and obviously particularly focused on mutations that we know have a biological impact, particularly on potential vaccination and reinfection. So we now have a systematic setup for doing that. I think one of the critical things is that the hypothesis is that these particular variants that have a large number of mutations, particularly immune escape, may well arise in immunocompromised or people with long-term infections. So we also try and sample those as much as possible.
And also, like the South African variant has been discussing a lot. Do you know the prevalence or distribution of the South African variant in UK or?
Yeah, so it's been detected in the UK, and it's now monitored on a day-to-day basis. And there's been, as I mentioned, where it's been detected, there's been mass testing. So they've gone in, if a location, if an individual is detected having it, they then go in and do mass community sampling of that area and then sequence to look at to look to see if it is actually spreading. We're also obviously our homegrown B117 lineage is also acquiring immune escape mutations, so we're obviously keeping track on those as well, and then looking for new variants. And we haven't. There's been a number of people in the consortium that developed tools, particularly looking for these. There's a tool called Polecat, which looks for the emergence of clusters that are starting to grow, take into consideration a number of factors. So again, those tools are publicly available and can be made use by anybody.
Okay, and so there's one more question. For the Kent variant, is it said that, the transmission is so fast and it binds easily to the kids more easily? Is it true? So like it does transmit. It's significantly better transmission, and you can see that wherever it's been introduced. I think probably some of the best data is from Denmark, where you can see that it got introduced in December, and gradually cases in Denmark have gone from being sort of 10, 20 to the point where now it's basically nearly all the cases, and you see that worldwide. I don't think there's any evidence for it that it transmits more rapidly in children. No, I've not seen any evidence of that. Okay. And also, if I didn't understand wrong, you are also doing sequencing for the people who has been vaccinated.
Yeah. So anybody who's been vaccinated, there's a cutoff applied. It varies slightly between 10 or 14 days post-vaccination. If you test positive, well, if you book a test and you've been vaccinated, there's now a system that pulls information from your medical records and ensures that your sample is then sequenced. So they've been doing that.
So sorry. Okay. So they shouldn't like reinfected, right? They are just vaccinated. They didn't have COVID-19 before, and
yeah. So anybody basically, if you've previously had a COVID test and then you have a second one, that sample will try and sequence that sample. If you've had a vaccine and then you test positive 14 days afterwards, we'll sequence that sample as well.
Okay. Thank you very much, Dr. Harrison. I'm not going to keep you a lot. I know that it's Easter time there, and you need to catch up with your family. So thank you very much indeed for having being with us today.
Thank you very much. It was a pleasure.
So I'm going to move to second speaker of today, Dr. Bas Oude Munnink. But before inviting him for his speech, I will give a brief introduction about him. Dr. Bas Oude Munnink is a postdoc researcher who focuses on the genetics of emerging viruses. He is working on preparedness for possible emergence of different arboviruses in Netherlands within the COMPARE, VEO, SHARP, and OFPA consortium. Since the SARS-CoV-2 pandemic, he has been involved in projects to monitor the evolution, spread, and transmission of SARS-CoV-2 through whole genome sequencing to enable fast genotype to phenotype predictions. Dr. Munnink, the platform is yours.
Thank you for your nice introduction. And indeed, I would like to talk to you today about what we do in the Netherlands for the genomic tracing. Oh, no, the screen is gone. For the genomic tracing of SARS-CoV-2 by whole genome sequencing. So this used to work, it's still working. Yes. So indeed, I'm working at the Erasmus MC, and we are currently sequencing a lot of SARS-CoV-2 whole genome sequences. And we want to do this mainly for whole genome sequencing for outbreak investigations. So after the first sequence was generated by Chinese collaborators and quickly shared already on the GISAID platform on January 10th, we were able to use this information to also develop whole genome sequencing protocols. And by now, over 800,000 whole genome sequences of SARS-CoV-2 have been generated and have been shared internationally. And as also said by the previous speaker already, a lot of sequencing effort has been done by the UK, but also several other countries really produced quite some sequence data. And in the Netherlands, we want to use whole genome sequencing to understand where the virus is coming from and how widespread the virus is. And next to that, we can also use it to see how different it is from what we've seen before, if it's part of an outbreak and if it's part of a potential transmission cluster. So to do so, we mainly use the Nonabortment ion sequencing. And the main advantage of this sequence machine is that it's relatively fast and it's fairly cheap. So this has been really promoted before already during outbreak-based sequencing. And examples of these are the Ebola virus, the Zika virus, and yellow fever virus outbreaks in Brazil, mainly, where they also use this fieldable sequence technology to generate whole genome sequences. And the main advantage is that it can perform real-time base calling, so it's really a lot faster compared to the more standardized Illumina machine workflows. So for SARS-CoV-2, we use an amplicon-based sequencing approach, which has been described before for the other arbovirus like viruses like Zika, yellow fever, and these kind of viruses. And we use we use 2 overlapping PCRs to generate overlapping amplicons of around 500 base pairs in size. And then we pull the 2 PCRs after the PCR step, and we sequence all different amplicons in one reaction. So after that, we of course still have a large part of the data analysis. So this is just an overview of a tool developed by the people from the UK, from the COG consortium and from the Arctic network. And here you can, in real time, monitor your sequence run. I will modify this script a little bit to automatically collect the data from the machine and transfer it to a server where we then can perform a real-time data analysis. So what you basically do is you try to demultiplex your samples, so you get your sequence tree divided per sample. You try to trim off your adapter primers, your adapters and primers. You align it to a reference genome, and we generate a consensus sequence with different depths cut off. And the main advantage of this real-time data analysis is that you can have a look at your sequence run as soon as you start it. So most of the times after 1 or 2 hours after the sequence run has actually started, we already have some information about which variants might be present in the data set. So after that, we try to reconstruct the entire genome. So we generate around 12 million sequence reads per sequence run. And this is around 10 gigabytes of data. So that's quite a lot of data which is generated all in all. And we've now performed over 200 sequence runs and generated around 2,000 sequences. It should be correct, it should be around 8,000 now. So we generate quite some data already. And especially in the beginning of the SARS-CoV-2 outbreak in the Netherlands, we could really use this for public health decision making. So we were able to use the rapid SARS-CoV-2 whole genome sequences to also use it for informed public health decision making. So we could see in the first phase, where we in the Netherlands still tested only travelers according to the WHO and ECDC case definitions, that we got our first positive case in the Netherlands on February 27th. And we got one additional case 1 day later, and we were able to generate the whole genome sequences of these 2 positive samples already 2 days later. So that's pretty fast. So within 48 hours after the first detection of the virus, we also had the first whole genome sequences of this virus. And we could see that immediately from the whole genome sequences already that they are most likely not from one source, and they are unlikely to be connected, but it seems to be that there were 2 different introductions at that time already. So during the next phase, we implemented the systematic sequencing during the exponential growth phase. So at this moment, we still try to sequence every positive SARS coronavirus 2 case. At that time, it was still feasible to do. But also for the diagnostics, there was already getting some problems that not all people could get tested. So in this phase, we had a specific focus on new cases with an emphasis on healthcare workers and severe hospitalized cases. So we try to, at least for the critical groups, we try to still do the diagnostic PCRs, but it was not feasible to test everyone who had symptoms. And we then observed also cases with similar travel history. We observed quite some diversity. So in the early phylogenetic trees, while there was still limited genetic diversity, we could already see some differences there. And at this moment, the sequence from the Netherlands represented around 27% of all the sequences generated worldwide. So in the beginning, we were really up to speed in trying to get whole genome sequencing ongoing. And then during the systematic phase, we also saw that, well, in the beginning, most cases could be linked back to travel history to Italy in this case, that also the virus could be detected in a lot of people without actual travel history. So it seems that the virus was already widely circulating within the Netherlands, and that these viruses were also pretty diverse. So we could see that there was the circulation of several sequencing types, which can be found in several clusters in the phylogenetic tree. And based on this genetic diversity, as well as the increase in the amount of COVID patients, and also the increased affected geographical areas, so we could find the virus basically in the entire of the Netherlands. This provided more support for increased movement restrictions. And then it was also the first time that actually phylogenetic analysis was shown in a briefing to inform the House of Representatives. So also then the government and the more broader public really got some experience with what you can use sequencing for. And that's now being more commonly being used for public health decision making, that they also want to have a look at the sequences. And they really see an added benefit of sequencing SARS-CoV-2 positive cases. So at the EMC, we also do a lot of outbreak investigations. So one of the things we want to determine if there's perhaps a geographical signature, but that's still very limited with the current mutation rates, in which it's possible to distinguish between different types and also different variants, but to have a real geographical signature, that's still pretty complex. But we also want to monitor significant changes in the viral genome to try to identify variants of concern or perhaps some other things where we should be careful about. And we do a lot of outbreak investigations. So we sequence a lot of samples from hospitals. So one of the things we did also in the early phase of the outbreak is to look in COVID-19 in healthcare workers in 3 hospitals in the south of the Netherlands, because there were some descriptions of quite some large hospital outbreaks. But we, in this case, used whole genome sequencing to try to discriminate between actually a non-communal infection and between an infection of just acquired from the local population. And we could see that there was limited spread within the hospital in these 3 hospitals at least, and it was mainly acquired from a normal population. We also do a lot of outbreak investigation in the field labs now. So we try to open up again a little bit in the Netherlands, and we try to experiment with concerts or some other big gatherings of people, and then we try to see if coronavirus spreads during these events, or perhaps you can safely have these kind of events in case you don't have any symptoms, and in case you have a negative corona test before you enter these concerts. We also do a lot of outbreak investigation in nursing homes, in slaughterhouses, in gyms, in schools, in the fruit industry, and also on mink farms. And I'll tell a little bit more about the work we did on these mink farms. So we already know that SARS-CoV-2 can be detected in animals. So it was first detected on a seafood market and a live animal market in Wuhan in China. And although they could find the virus here in environmental samples, they could not find one particular animal species which tested positive for SARS-CoV-2. And also animal experiments and in vivo experiments just happening in nature have shown that cats, ferrets, hamsters, macaques, and fruit bats are susceptible for SARS-CoV-2, among many other species now. And we've also seen that dogs, cats, lions, tigers, and minks can be infected with SARS-coronavirus too. So after we got a report of an increased mortality at the mink farm in the Netherlands on the 19th and 20th of April, where around 2.4 to 1.2% of the animals died, they first tried to test for the more common mink pathogens. But when these were all negative, they also decided to test for SARS-CoV-2. And they could find that these 2 farms, almost all animals were positive for SARS-CoV-2. And at the moment, we don't have any mink farming in the Netherlands anymore, at least not industrialized. But before that, we saw that the virus quickly spread from farm to farm. And there were 67 mink farms in the Netherlands which reported SARS-CoV-2 infection in the end. And these were all culled just to prevent the spread of SARS-CoV-2 any further. So when we then look at a big circular phylogenetic tree and we compare the mink farm related sequences to all Dutch sequences which are produced at that time, we could see that there was relatively a lot of divergence compared to what you see in the human population. So there was really active evolution of the virus, or a mutation of the virus in mink farms. And we could also see that there were only 3 main clusters which could be detected in all of these 67 mink farms. So it seems that all of these mink farms have some sort of connection to each other because they are infected with more or less the same virus. And in the first 16 mink farms, we also actively looked at farm workers. And here we could see that 68% of all tested mink farms tested positive for SARS-CoV-2 either by PCR and serology. So it seems to be quite a risk factor to work on a mink farm where SARS-CoV-2 is circulating among these animals. Although they try to implement very strict hygienic measurements, so they had to wear gloves and face masks, but apparently this did not help against getting infected by SARS-CoV-2. So also in these mink farms, we found the first proof of mink to human transmission. So this is based on 2 different methods. So not only whole genome sequencing and phylogeny, but we could also see that the employees got infected after SARS-CoV-2 was first identified in minks. So we could see that 2 weeks after the initial infection of SARS-CoV-2 in minks, we could see that the mink farm employees also tested positive. And then whole genome sequencing showed that they were infected with the same virus which were circulating in the minks. And we have to say that most probably not all human cases are mink to human transmission, but most of the times they also live in the same house and it's a family business. So it can also occur that perhaps the virus spread from mink to humans once and then it spread subsequently within the family. So we also try to look at early detection of the virus. So we actually got a system in place that every week all dead minks which are reported or observed at the mink farm has to be sent in for early warning signal. And these will then be tested for SARS coronavirus 2. And we can see that we made a time resolved tree, and then we can see that we were pretty early in detecting the virus at these different mink farms, because when we look at the time scale, the most common ancestor of these viruses most of the times only a few weeks before detection. So we were pretty convinced that we were quite early in detecting SARS coronavirus 2 outbreaks on these mink farms. But there are some exceptions. For instance, for mink farm number 6 here, we could see that there were already quite some divergent viruses circulating. So it can be that this one was missed in the early detection system. But at the moment, the mode of transmission on these mink farms is still unknown. So we tried to make also a more structured transmission model using genomics data and also the distance between the farms and if they have any common features. But at the moment, we still could not find a real mode of transmission. So we did sequence also cases from the surrounding of the mink farms, and these were not related to what we see happening on these mink farms. But in the phylogenetic tree, we could see that there's mainly one big extension of one big cluster. So there seems to be some sort of link, but we were unable yet at the moment to find this link and to see how the virus spread from mink farm to mink farm. But that's something we are still looking into. And as Ewan already also mentioned, at the moment we're also busy in trying to look at the varieties of concern. So there's the B117 variant, but also the B356 variant in South Africa. And then there's also still the P1 variant from Brazil, which are shown to be perhaps more deadly, but at least more easy to transmit. And one of the hypotheses how these variants occurred, because in the UK they sequence a lot. So it's, I heard just now, up to 9% of all positive cases are being sequenced. And then all of a sudden they detected a variant of concern, which has 22 nucleotides difference from what you've seen before. So we're still trying to figure out how this virus got introduced. And one of the hypotheses is that it might have occurred in an immunocompromised host. So this is a paper by Cho et al. Who followed one person in time. So this particular person was followed 152 days in time. And he had an immunodeficiency, so he could not clear the virus, and the virus remained to be present in this individual. And then when you look at the whole genome sequencing of this individual, you can find certain mutations which are now also typical for the varieties of concerns, which we are starting to find now. So also in this particular individual, we could find the E484K mutation and the N501Y mutation, which are also now pretty often found in the newly emerging variant of concerns. So it might be that these variants occur in immunocompromised hosts and then somehow find a way back into the general population. And also in the Netherlands, we have sequenced now 3 immunocompromised patients, which have been followed in time. And also in these 3 patients, we start to find back the same mutations that have been found before in this immunocompromised patient, which can also be found in the variants of concern. So this is also something we are trying to follow up and trying to see where these variants occur and how we can, in an early phase, try to prevent the spread of these viruses. But also another way of infecting or generating these variants of concern might be that we don't sequence in very, how to say this, we sequence a lot in Europe and in the UK, and in the US also a lot of sequencing is being performed, but there are also some countries in which less sequencing is being performed. And one example is this is, for instance, Suriname. So that's also why we went to Suriname a few months ago and tried to do the sequencing there in the field. And there we could also find SARS-CoV-2 variants, which had quite some mutations already compared to what has been described before. So also here we see up to 15 nucleotides difference from anything what has been sequenced before. But I have to say that in this case, we did not find any mutations in the spike protein. So that might be one reason that it might be more likely that the variants of concerns are actually accumulating in immunocompromised patients. So at the Netherlands, we're now actively screening for variants. So we don't do it only by whole genome sequencing, but also by performing a specific PCR. So there's a PCR which has been developed to detect the 484E or 484K variant, and then the 501N and the 501Y variant. And this is being done by a real-time PCR, and by performing a melting curve later on, we can distinguish between the different amino acid residues which are present at these sites. And this can also be used to detect the deletion of amino acid 69 and 70 in the spike protein. And when we use this test to look at the spread of the B117 variant in the Netherlands, then we got a picture like this. So here below are the different week numbers of 2021. So we could see that before 2021, this variant was not present that often. But when we look into the 7th and 8th week of 2021, we can see that the virus is, the prevalence of this virus is really increasing. And in some cities, it's already increased to 70 to 80% of all cases, which are now the B117 variant. And then there are also still the B1351 variant, which is the virus which can be found in South Africa, and the P1 variant, which can be found in Brazil. We could also see that these viruses start to pop up in the Netherlands. So for the B1351 variant, we already have around 100 cases, and it's increasing. So it seems that this variant is also already rapidly spreading within the Netherlands. And also the P1 variant at the moment can be found only occasionally. But in case we found these variants, we also still try to increase the contact tracing and trying to prevent further spread of this variant. So just to end with some sort of discussion, I think it's important to keep track of how the genomic diversity of SARS-CoV-2 is at the moment. And it should be combined with epidemiological data, but it can be very sensitive to share metadata publicly, because of course it's important to guarantee the privacy of the patient. And it's important to track and control potential animal reservoirs of SARS-CoV-2. Because for instance, now we've also seen that mustelids are sensitive, but we've also seen that mice might be infected with the novel variants which are occurring now, and perhaps they can also play a role there. We also think that more and more variants of concerns and variants of interest will emerge in the future. So therefore it's important to have a fast genotype to phenotype prediction, So you can, in early stage already, try to identify these variants of concern and try to prevent the further spread of these variants. And what we are now actively doing is to monitor potential immune escape variants after vaccination, to also see if perhaps some variants will show up there. And that's what I wanted to share with you today. And I would like to acknowledge all of these people who contributed to this research.
Thank you very much, Dr. Munnink. Actually, worth to mention that on behalf of Cyprus team, Cyprus COVID-19 team, I would like to thank you for accepting our collaboration, and then you sequence some samples for us. And seriously, please accept our special the thanks to you. I hope we can sequence soon the rest one. I hope it will be successful. I know that you are very busy.
Yeah, indeed. I forgot to mention that in this talk, we indeed received some samples from Cyprus, which we also have sequenced. We also try to upload them as soon as possible to GISA to share them also with the broader community.
Yes, thank you very much.
And we'll process the other samples you sent to us and try to see what we can find there.
Thank you very much indeed. There are some questions. If Dr. Harrison will be here, it will be nice to discuss, but I think he's not here. He has to leave us. So thanks for the great talk. What would be your suggestions for the countries who are just starting to set up their sequencing facilities for tracking SARS-CoV-2 evolution locally?
It's very important to get the sample flow organized properly, because we also find sometimes that it's difficult to actually receive the samples and trying to keep them or to receive them as soon as possible. So that's essential, I think, also the administration and trying to keep track of all these different samples. And I think I would advise to start with the Nonabortment sequencing platform, because that's relatively cheap and quite easy to implement. But it's also difficult to do the data analysis later on. So it's not just the sequencing, it's also quite some data analysis, which can still be quite challenging. And it's also something we discovered in Syria. So we actually went there and performed the sequencing locally, which went fine. But after we went back, the sequencing stopped, because they didn't have any people with the expertise also how to deal with this large amount of data which you generate after next generation sequencing.
Yeah. Well, thank you for the question, Dr. Battal. Also, she is one of our our team member. There's another question from Turkey, Professor Temel, about the deaths after vaccination. If you're aware of, is this person sequenced if they have any common variants? Have you done that kind of experiment? First of all, I have to ask this.
So after vaccination, we have not found any individuals yet who got infected by SARS-CoV-2 and have died. But we do see sometimes infections after they got vaccinated, and most of the times then we see less severe disease. So that's an advantage, and it seems that the vaccine is really doing something also in these individuals. We do sequence them after they get vaccinated and they do get infected. And at the moment, we see wild-type viruses just showing up, but also some of the different variants. So we see a lot of the B117 variant now showing up after vaccination. But that's difficult to really say something about it, because that's also the common variant which you see now in the general population.
Is it the UK variant now you are seeing more commonly in Netherlands as well? Or?
Yeah, it's now 70% up to 80% of all cases are the UK variant.
Okay. There's two more questions. Thank you for the presentation. You have mentioned that some variants may have different melting point, and that's how you distinguish them during PCR. Can we say that variants are lead to massive structural changes for the virus since they have different melting points?
So this is a particular assay which has been developed by a company, and they changed the annealing temperature of the probes. So they can play around with the length of the probes and thereby change the annealing temperature. So it's actually, it doesn't cause any changes on the structure of the virus, or it might. But in this assay, you want to detect it, you will just play around with the length of your probe, and therefore you get different melting temperatures.
This is actually what is very widely has been using in the molecular diagnostics as well, actually, the technique. And one more question. Thanks for the insight and the presentation. Was there comparative sequencing variant with the SARS-CoV-2 emerging in other countries? If yes, how can we use this data to control the spread?
I'm trying to understand the question. So there was a sequence variant which also emerged in other countries. So we're now also actively talking to WHO, trying to also monitor these variants, because there are a lot of variants of interest at the moment. And it's very difficult to distinguish variant of concern with variant of interest. And then indeed, one of the criteria there is that a variant should be present in more than one country and should be spreading in different countries before we can really classify it as a variant of concern. So we do use the epidemiological information and data from different countries to come to that decision.
Okay, thank you very much, Dr. Munnink. There are some people are thanking for your talk. So great things. There are some great things. Well, I think we are done today. So I'm really thankful for your time, for saving us for today. And I hope we will be seeing you in soon. So maybe next time we can invite you all in Cyprus and you can enjoy the weather as well after COVID-19, obviously. Okay.
That would be nice. Thank you for your invitation.
Thank you very much, Dr. Munnink. Have a nice day.
Bye.
Bye.
Catching a bus, never nothing when it's real, yeah it does. I'm flying above.
Yes, today we have finished our talks, and I hope we're going to see you in next time, and maybe face to face without COVID-19. See you later.
Catching a bus, never nothing when it's real.